let's go ahead and get through announcements and the general introduction. So I'm Wendell Jones. I'm president and executive officer of the MAQC Society. We're glad to have you join the MAQC Society sponsored webinar on the SEQC2 series, looking at current research and papers that are about to be published in various journals such as Nature Biotechnology, Genome Biology, and Cell Genomics. Today, we are glad to have joining us Dr. Chris Mason from Weill Cornell Medicine, who's going to give us a great talk on the Epigenome Quality Control Project in just a moment. We also have a presentation tomorrow from Dr. Jonathan Fuchs, who is also in Chris Mason's lab at Weill Cornell on more of a metagenomics field, looking at both benchmarking human and bacterial sequencing, multi-platform and multi-site in different contexts. And then next week, we will finish up our webinar series by looking at germline, difficult germline variant calling in different contexts by Dr. Wieschow at the FDA. And so let me introduce Dr. Mason now. So Dr. Mason uses computational and experimental methodologies to identify and characterize the essential genetic elements that guide the function of the human genome, with a particular emphasis on the elements that orchestrate the development of the human brain. His lab at Weill Cornell creates detailed cell-specific molecular maps of genetic, epigenetic, transcriptional, and translational activity, creating a draft of the molecular recipe for the creation of the brain. Chris also develops methods to detect, catalog, and functionally annotate variants in the genetic pathways that control developmental processes and how they are perturbed to create disease. His research aims to understand the functional elements of the human genome well enough to enable, eventually, the ability to repair, engineer, or fortify the genetic networks within human cells. Chris, we're glad to have you here, and take it away.
Great, thank you so much, Dr. Jones. Really pleasure to be here, and good to see some familiar names and some faces as well. It's normally we'd be doing this in person, of course, but today we're doing it with Zoom, as we've all gotten used to. So let me share my screen here and close some things down, and then hide this. Okay, and then let me know if you can hear and see me. Okay, see the slides okay, and hear okay?
Yes, I can see the slides. Looks great.
Great. Okay, wonderful. Well, good afternoon, everyone. I'm going to take you on a little bit of an adventure in epigenetics today, and basically some of its relevance for different clinical contexts, some work we've done with the SEQC2 EpiQC project, which is about quality control and figuring out ways to do epigenetic measurements in a better fashion. And also it's some other fun things of where we're using this for some funky space epigenetics as well. So I'll start with a background of that, as I think really familiar with everyone here is that, almost every medical school, Cornell included, has its own precision medicine institute. But what I want to think about is to think not just about measuring when something goes wrong, what you should do for an action, but what if we could be more, not just precise with our medicine, but become more predictive and see what is the trajectory that you have for any molecular change. It could be genetics, it could be epigenetics. I'll talk about both of them today. But it could be post-translational modifications. It could be a metabolite that's changing. That's a biomarker, an indicator for potential disease. All these facets of biology can, to some degree, potentially be informative. And so we have an initiative at Cornell that's actually about quantitative prediction, is a way to actually see how far we can predict using new machine learning and other methods to see where are you going essentially in your medical trajectory, not just how did you get there. And you can see here we're looking at just a screensaver, but anyway, it looks very exciting when this was first announced. But the model is basically, how do we think that not just when you're healthy or if you get disease, have precise medicine, but get more predictive about how do we get you back towards a healthy state and avoid disease in the first place. And so this is, not just a goal in our lab. This is part of large scale efforts across the country, including the All of Us program, where Cornell is actually one of the collection sites. And so this is people donating blood, saliva, stool, a lot of other features to better understand general genetic variation and using it for precision medicine. I just got off a call this morning with the Million Veterans program. Actually in there, they're also doing whole genome sequencing to have genome guided care, but they're expanding very soon into epigenetics as well for a lot of their veterans. And so this is, how do we safely take care of people who've gone to war? How do we guide pharmacogenomics approaches? In general, just understand their health and well-being. And that's just in the US. If you look globally, things have really changed a lot. So, this idea of population scale sequencing and genomics and epigenetics, or PopGen, is really greatly broadened in the past 2 to 3 years. So now it's really global efforts. Many countries and states having their own infrastructure and testing paradigms to really get part of the EPIC records, part of medical care. The genomics is becoming a key part of it. And actually, one of my more recent favorite examples, again, most of these programs are precision medicine looking backwards, but there's finally large shifts to look more predictive medicine, or to look forwards. For example, the National Health System in the UK is partnering with Grail to actually offer basically cell-free DNA sequencing to 165,000 people this year that have no sign of disease. And if it looks like it's successful and help people avoid cancer early, then we're going to expand it to 1 million people in about 3 years. And what's interesting about this is the cell-free DNA are fragments of DNA that are floating in your bloodstream that come from all the cells in your body. But if you have cancerous mutations, that could be a way to predict, are you getting cancer? Is it a really early warning sign system for cancer? But it's never been tested at this scale. And so if this really does come to pass, this will be a wonderful example of the power of kind of predictive medicine. But what's interesting about this is not just that you get, of course, genetic changes, what are present there. We also get, epigenetic changes. So the fragments of DNA, as they come from different parts of your body, will indicate which tissue contributed that signature. So when you sequence the DNA, if you look at the epigenetics, you can also tell from which tissue it likely came in your body. And so we're doing this also with urine sequencing. Just last week, we're doing a project on this and some new data. So, it's not just about what mutation you see, it's about what tissue is contributing those mutations. So I'll talk a little bit more about this later. But for a little bit more backdrop first, is, also one thing we know that changes over time is the epigenetics essentially will drift, but also, which I'll show you, but also telomeres shrink over time. That's something that we lose with age. Another thing that we gain with age is mutations. They're called somatic mutations, what are called clonal hematopoiesis of indeterminate potential, or CHIP, is something else that accretes with time. There's one other thing that changes with time, as if you don't already know, is basically this epigenetic layer, which is the regulatory layer behind DNA. And in particular, one of the particular modifications that we focus on is 5-methylcytosine, or 5-MC, really just a small methyl group to the C of your genetic code here in black. The epigenome marks here in red show you can have these very small chemical tweaks that can control when and how your DNA is packaged, utilized, and regulated. So this is kind of part of the tweaks and the levers of biology. If DNA is the hardware, epigenetics is often called the software. And we know that it's not stable. So if you look at what's happened, for example, in identical twins, if you look for different methylcytosine sites at 3-year-old identical twins versus 50-year-old twins, and this Fraga et al. Paper, you can see that you get a lot more differences, meaning their epigenome have drifted over time in those that are 50-year-old versus 3. And so this is actually so consistent. This led Steve Horvath in 2013, after Hannon did it in 2012, to basically come out with markers for predicting your age. He said, well, if that's true, then we could actually tell how old you are based on your epigenetic age. This was validated by many other groups, ourselves included, since then. And you can even see if you take induced pluripotent stem cells, you can reset your chronological age back down near 0 because you've reset all the epigenetic marks. And so, this ability to see these changes is, still being perfected, but actually works pretty well. We're even trying it right now on a crime scene sample that I'll be talking about later today with John, Dr. Fuchs, whom you'll hear from next week. But so this is kind of an exciting development and application. But the other thing about epigenetics and DNA methylation is what's amazing is we're still discovering things. So for example, just from earlier last week, new enzymes are being found that can basically protect and maintain DNA methylation valleys. So for example, most of the genome is hypermethylated. You see a lot of it has a lot of pink here. And there are these valleys of demethylation that appear, often where you essentially have activated genes, but not always. It can be, a little bit, it can be more complicated in some degree. But you can see we're still finding, these tools that are controlling what is protecting these valleys from getting hypermethylated. And so that's in sort of normal development. But if you look in disease, we also see the significance of DNA methylation, in particular in diseases like colon cancer, glioblastoma, and also acute myeloid leukemia. Some of the most recurrent mutations in leukemia are epigenetic modifiers, including things like the TET enzymes IDH and DNA methyltransferases, DNMTs. These are among those samples most commonly mutated. And so what's interesting about this, when they get mutated, what they do is they disrupt what is a normal occurrence inside the essentially the cells, where mutations lead to impaired hydroxymethylation development and increased DNA methylation. Because if you essentially have a mutant IDH1 gene, it basically leads to 2-hydroxyglutarate, which basically blocks the functioning of these enzymes. So you start to just accumulate hydroxymethylcytosine instead of methylcytosine. So it inhibits the normal processing of cytosines that occurs across the genome. And so mutations in these genes, these isocitrate dehydrogenase genes, you can see IDH, are really key to driving certain leukemias like GBMs and also leukemias. And they're not just driving the cancer, there's a much worse prognosis. So if you actually do have some of these modifiers, it's similar to unfavorable cytogenetics in this regard. So we know that these are poor markers as well. So over the past few years, we've developed a number of tools to look at some of these questions in DNA methylation. So one is called MethylKit, which is very commonly used. There's also one called EDMR, and there's a number of other tools. At one point, we thought about calling this tool MethLab, but we thought that'd be a poor choice for a algorithm name, so we didn't call it that, as you can see here. But we've now released over the years a variety of different tools which I'll show you some today, that look at clonality, that look at DNA methylation, and also even look at Oxford Nanopore data. And we've developed these tools because we want to look at cancer samples in normal, but also, as I'll get to with EpiQC, figure out the best ways to actually call modifications. So one interesting example with longitudinal changes, again, is from a patient we looked at who actually worked and cleaned the nuclear reactors at Chernobyl after the accident in 1986. And he unfortunately developed cancer, developed leukemia, but we had 5 time points to look at both genetic and epigenetic shifts in this patient, whom we called Chernobyl man. And we could actually see here that in this regard, the number of somatic mutations increased more over time, but it was the epigenetic changes that were much more pronounced early in the development of the disease. And so this led us to say that it seems like in this case, this might be driving the disease to go faster. In this case, this was just the over amount of epigenetic heterogeneity. And the way that we measured that was actually kind of a new method. So in particular, we looked at epialleles, which is if you take all your sequencing reads, and I'll come back to this shortly, bisulfite sequencing reads. Essentially, if you see adjacent CPG sites, you can get a pattern within this one read. Basically, the information is phased within one read, one molecule, and that lets you gauge essentially the clonality that was contributing to this epigenetic signature, or we call the epiallele patterns. And here from diagnosis to relapse stage, we can see these shifts there, these loci that seem to be the most dramatic and changing during therapy. And so we looked at basically diagnosis versus relapse, or different versus normal bone marrow to see what's the E loci per million loci sequence, or this degree of epigenetic change. And in particular, we noticed that there's a wide range of variations at diagnosis versus normal, relapse versus normal. But if you do relapse versus diagnosis within the same patient, sometimes you'll see no epigenetic changes, but you'll see genetic changes, and you'll see the inverse. You'll see no genetic changes, but you'll see tens of thousands of epigenetic changes. And so what's interesting is using this as an idea of like, does this have a prognostic relevance? We basically look to see if we see this selection of a specific clone, does it matter for the outcomes? And this was from our first paper in 2016 that indicated the answer was mostly yes. It like basically meant that there were separate paths for epigenomes and genomes in cancer evolution. But this has been followed up by Dr. Lee, who now has her own lab, actually has been following up on this in the past few years, and showed again with 2 different validation cohorts that we can see the same thing. So when you have all this increased epigenetic heterogeneity, we can see that you are, basically, it gives you more essentially escape hatches for the cancer to evolve. And so we can see that this is, essentially a really poor prognostic marker. And so this led to, questions of just, we don't just want to stratify patients, we just think, well, can we target this aberrant methylation in a better fashion? And in this regard, we've actually published already some work of using different inhibitors that either inhibit FLT3 or IDH from Ambit or Agios, 2 different companies, or comparing it to azacitidine, a hypomethylating agent, and seeing, at least in mouse models, what if we do some of these combination therapies and see if we could actually target these sites of aberrant hyper or hypomethylation. So in particular, when we have these differentially methylated cytosines versus the vehicle of hypomethylation in blue or hyper, you can actually see here, we can actually really start to pull back and get more hypomethylation, essentially when we have this combined treatment here, or get the balance back, right? Or if you look at this here, we call these kind of melt curves. If you see versus normal, we want to actually try and bring it back down. If you do just a vehicle, isn't really no treatment, but if you do combined treatments, it starts to get it back towards the zero, back to normal. Basically, 0 represents you're back to the normal methylation state. If you look at this by colors, what's the percent methylation difference from normal? We can see the combined therapy did an extraordinary job of getting us back towards a normal state. And so, that gives you a little some of the clinical context and some work we've done and thinking about where is DNA methylation relevant? How do we target it? I'll come back at the end about some newer epigenetic CRISPR-based methods that are just coming online, which are pretty cool. But at least in terms of combination therapies of hypomethylating agents, we know some of these actually can be quite effective in combination therapies. So that's at the applications, the detection, but it really begs the question of, can we detect them better? And what are the methods that are out there for DNA methylation detection? There are many, like with many molecular assays, there's a variety of ways to just answer this question of find me methylcytosine. I'll highlight a few of them quickly, and then we'll dive into the EpiQC. So one of them that's been around actually now for 11 years is single molecule real-time sequencing, where you have PacBio sequencing with the DNA polymerase at the bottom of the pore, and you can watch the polymerase as it essentially goes through the template strand, makes a complement of it, and you watch in real time the kinetic activity of this DNA polymerase kind of squeezed at the bottom of a well, active and churning through a template strand. And so what's cool about this is you get, of course, single molecule sequencing, you get ACGT, but you also get the kinetic information from the polymerase, which itself is an informative marker of what kind of state the bases were in the template strand. So in particular, if it's methylated, you can see it changes the incorporation speed of the polymerase that can indicate whether it's methylated or not. So it's not quite perfect, but it can find methylcytosine, hydroxymethylcytosine. There's a slide from Jonas Korlek. You can see here that the kinetics can detect maybe other DNA modifications. That was kind of cool. We've also worked with PacBio historically that you can, instead of putting a DNA polymerase down there, you can put a reverse transcriptase, and particularly an HIV reverse transcriptase at the bottom of the well. And you could even do this to look at, say, direct RNA sequencing. In this case, you could say, oh, I have an RNA template and a DNA primer, and watch cDNA synthesis occur in real time. And here too, even if you have something at the bottom there that's regular DNA primer and RNA template, or if it's methylated, the methylation will actually change it even for RNA sequencing. And so we published this back in 2012, showing that this is a way you can actually detect modified RNAs as well as DNAs on the platform. But unfortunately, it mostly stalled there. PacBio hasn't developed that platform further. It was very tough to get to work, although it's possible. And around 2014, Oxford Nanopore came onto the scene, and they had different versions of their technology, the MinION, the Prometheon, the GridION. For a little while, they debated between which method they should use, whether they should have an exonuclease sitting at the top of a pore and chop off one base at a time, or to pull through with a motor protein one strand and try and infer the bases based on the changes in the electric current. So you can see here, they ended up commercializing StrandSeq. Actually, historically, Illumina had invested in this one, but they went with this one. And basically got this out to market to say, okay, if we can pull this through, we can actually convert this current into bases. And if it's modified, maybe it's slightly different. In particular, if it's methylcytosine, could it be different? So now fast forward to today, there's a variety of platforms, and I'll present to you actually a whole bunch of data that's just in the past few months that we finalized that's going into a paper that's probably being resubmitted as part of the SEQC to FDA package today, the last edits, or tomorrow, that details some of these latest developments. But basically, what we're trying to do is convert this raw signal, the squiggles, into cytosine and methylcytosine. And it's interesting to note, this has a lot of applications. So this is not just for thinking about, sort of basic clinical samples that are happening here, but this could be used anywhere, because the MinION, as I just showed you here, is very small. It's actually the very early version of it. You can use it in all kinds of spaces. So for example, we use it in a parabolic flight simulator here with Andy Feinberg. Just here he's using some of the first zero gravity pipettes he was trying that had positive displacement. You can see what goes wrong if you're in a flight simulator where you get a little bit of zero G. There's people that are behind him right now puking because of the change in their stomach, and you can see a little bit of tubes flying around. It's not really the best place to do science with all the tips there, but he kept his spirits up and maintained focus, got the flow cell loaded on the MinION. We showed that it was possible to do sequencing in zero gravity, but it was a little bit different. If you look at the current in picoamps of the flight sample versus the ground, they were different if you look at each K-mer. So this tells you if you're trying to find slight tweaks to your current to tell you whether it's methyl or hydroxymethylcytosine, you have to kind of really normalize the baseline. This is the hidden Markov model of the baseline model, and you can see the differences between some of our tests here. But nonetheless, we were able to show that it's possible to look, essentially, it's ready for liftoff. This works actually in zero gravity or 1.8 G or 0, 1.8. You can see that we actually normalized it and got it working. Eventually, this became the protocol that Kate Rubins took up to space and got sequencing in space working. And she's actually, just doing some more experiments as we speak, and is coming back to Earth in about 4 days after her second mission. We did show it's possible to sequence in space. This was really huge on Twitter. There were 4 people who retweeted it, very big, very big. And it's also showed that, we this is the first genome that was sequenced and assembled not on Earth, but also afterward we've published the first space epigenome. So this is kind of single molecule detection of N6 methyladenine, which I'll talk about this now at great depth. So in particular, these are new methods that we have, which is great. It's cool. We can use them in space. We can use them on Earth. We can use them on cancer. But we need, on Earth and space, they need new standards, right? And we don't just want these to be, vague standards. They have to be really reproducible, rigorous things that we can use across site, very MAQC, focused in terms of what we were expecting. And we don't just want to have meetings just to have meetings for no reason. This is a sign I have up in my office that, if you're lonely or tired of working on your own, you can hold a meeting. We don't just want to have meetings to be the practical alternative to work. We'd like to actually use these things in different products and different projects and different applications. So in particular, this has been a collaboration with the FDA, with NIST, and with the follow-up, also some work with the ABRF NGS phase 2 study. All of these are which now almost complete and slated for publication this summer if all goes well in Nature Biotechnology and Cell Genomics for this final phase. And really what I wanted to do is the SCQC1 looked a lot at RNA sequencing, and SCQC2 really did a deep dive with the FDA and a lot of colleagues on this call on DNA sequencing. And as I like to say, we really wanted to bring sexy back like Justin Timberlake did. So when we first proposed some of the ideas around this, we said, well, let's see if we could look at all these modifications in these samples, like methylcytosine, hydroxymethylcytosine. There's allegedly 4-methylcytosine in some specific species. And could we compare it to like Promethean data, PacBio, other arrays, different platforms, look at Earth and space potentially. And we wanted to, again, use these samples that are really well characterized. We know the genome well. There's also a really nice repository, a Git repo for all the different Jupyter notebooks and sort of tools and algorithms that are used. And these are well established cell lines that we know a lot about that other people can order from. So this is from a previous paper where we characterize them genetically. But what we also want to do is take the exact same samples and run them through sometimes multiple labs and multiple protocols and see what we could find. So basically, some of the methods we wanted to use included, as I alluded to before, bisulfite sequencing. And in this context, what you do is if you take basically, if you treat DNA with bisulfite, any cytosine that doesn't have a methyl group, that's just a naked cytosine, will be deaminated and then converted to uracil. And then after PCR, that U becomes a T, as it's read by the polymerase. Where if it's a methylated cytosine, it stays as a C and it won't get converted. So you can see it's kind of schematic here. And there's different kinds of methods, just regular whole genome bisulfite sequencing. One's called TrueSeq or a cell NGS methyl-seq, where you have a bisulfite conversion and then adaptase and PCR amplification. TrueSeq is bisulfite priming, end tagging, and then amplification. And SPLAT is splinted adapter tagging. What this means is you take the sheer DNA, you bisulfite convert it, you do a ligation to the 3' end, then the 5' end, do PCR amplification, and then you can actually sequence this product too. And this is a great slide from Jessica Nordlund, who's a key member of the EpiQC. So these are some of the, all these though involve, you can see up here, bisulfite. It's really harsh. It beats up the DNA. It basically, it does at some degree of fragmentation all on its own. And it's just generally a bit harsh, I'd say. And so that means your fragments will get shorter the longer you treat it and get beaten up. However, we did chat with New England Biolabs when they were first developing their protocol called EM-seq or enzymatic methyl-seq, where it uses, instead of bisulfite to do the deamination reaction, it uses a collection of ApoBec enzymes, which deaminates 5-methylcytosine, 5-MC and 5-HMC, hydroxymethylcytosine and methylcytosine. And so basically, to separate those two, it uses TET2 to actually modify 5-MC to HMC that won't be substrates for ApoBec. So in theory, it gives you better accuracy for finding just HMC. And so what we want to do is take, these protocols and then also compare it to Megalodon, which is built on a base caller for Oxford Nanopore that does base calling, reference mapping, and also base modification calls. And so in theory, this should work well too. And so we put all this together into the EpiQC, which is now, you can see here, the preprint is here, and it's also in review at Cell Genomics and has very good reviews, we think. So we're hoping this will be coming out this summer if all goes according to plan. And again, you'll hear actually Dr. Fuchs, who really fearlessly led this project of a large group, is talking next week. So I'm here representing mostly his work as well as many other people's work on this list. So, okay, so what does the data look like? We ran basically each one of these samples. This is, the 12878 sample, the Ashkenazi Jewish father, mother, son, as well as the Chinese father, mother, son. Again, these genome in a bottle samples, well characterized, well established. And we did EM-seq, methyl-seq, split, true-seq, true-methyl, and nanopore sequencing, essentially across all these samples. And did them in some cases across replicates for some of these protocols. Not for all protocols, but for at least for some of them, to get a sense of interlab variability as well. So what we observed is actually some intriguing things. So particularly, if we take a BAM file that's really deeply sequenced, we did, maximum depth, 10x or 20x coverage, and then did a lot of downsampling. And we could actually see the downsampling and the bed files, like what we're getting for the actual, percent methylation. You can see is 100% methylation or 0% methylation, and all the pairwise comparisons. You can see it's a pretty tight correlation in general across all of them. No, it's not, not perfect, but it is writ large across each one of these, fairly comparable. We then wanted to see, okay, if it's overall highly correlated, does it, what about inter-individual assay differences? So for each CPG in the genome, how often does one assay pick it up versus another? Do two of them get it or only three? So then we did this kind of basically these different plots that would show is something detected in say 6 out of 6 platforms, 5, 4, 3, 2, 1. So basically the red at the top here means that regardless of kind of the coverage, you see the different coverage thresholds at the top, and the size is the scale of the number of CPGs that are in that grouping. You can see that, if you have more red, that means that protocol has more assay specific methylation. Now, it doesn't mean they're not real. It's more likely that they're not, to be quite honest, but it means that, these more red is generally bad, more purple is better, meaning it's what it's calling as something that's present is generally picked up across all their platforms. And you can see here, Nanopore actually does quite well in that regard. The other thing we wanted to look at is, okay, that's this kind of intra-platform uniqueness or comparisons, and then also how do we gauge this relative to the coverage overall across like the genome, and what's the percent methylation that we see? It often across all samples you can see would dip down at the transcriptional start site across all platforms, which is what you expect. This is one of the telltale signs of where is a transcriptional start site, as you see this beautiful thumbprint of a demethylation that otherwise the genome is mostly hypermethylated. You can see it consistently drop down. But what's interesting about this, when you zoom into this plot here, you can see it's not exactly the same, right? You get a little bit of wiggleness that's happening here. In some cases, some platforms actually decrease a little bit of their coverage right at the TSS, more than others, and other ones have a bit more noise that's present. But for the most part, the signature was well maintained. And so that was across these platforms. The other thing we want to look at is how does the newer platform or newest entree into the market is the megalodon called Nanopore 5-methylcytosine calls compare against whole genome bisulfite sequencing methods or other methods like this. And you can see here this correlation matrix on the bottom here is it's at about 0.92. Other ones are about 0.93, 4, or 5. So it's actually fairly comparable to essentially other methods on the market that are currently used. And in this regard, this is at 20x coverage. You can see here the R squared's looking pretty highly correlated. So it means it's picking up a lot of the same things at about the same levels. We then want to look at this at you can see here at 20x compared in Nanopore versus the other methods, it looks pretty good. And if we compare to some of the focus regions for what's called the EPIC capture bisulfite sequencing, even at 10x coverage, actually, there's generally pretty good correlation between them. So you will get some odd outliers. But overall, this shows that we're actually quite pleased that Nanopore has been talked about for really almost a decade as being a way to actually give you a lot of this kind of information. And it looks like finally now, coming back to this slide, that it's actually doing a pretty good job. It actually, I'm quite elated because we've been talking about using this to get genetic and epigenetic information for a long time. And PacBio's coverage is still a little bit high and also is a bit more expensive, but Nanopore looks like it's really coming into puberty as a platform, if you will. It's really growing up. So really excited to see that in terms of what we see in these data. And that's also going to be in the paper in the final revision that's going in and hopefully published again this summer. Okay, so I want to switch gears a bit to say this is what we've seen a lot for the intersite variability and some of the methods for the EpiQC. I want to close for the last kind of section of the talk about, well, I couldn't resist. What about in space? Can we use some of these methods to look at variability over time and other sort of features of spaceflight? So as most of you know, have heard me talk about probably, we looked at Mark and Scott Kelly. They're identical twins. Scott went in space for a year. His brother Mark is now a senator. We will have the ability to see what happens to him, to his epigenome after he's a senator. We'll be continuing to collect blood from the lab. So we'll see what being in the Senate does to your epigenome in about 3 or 4 years. But to go back to the previous study, the twin study, we looked at essentially everything that we could about the biology of the samples because we didn't know what would happen in spaceflight. So this included DNA methylation, hydroxymethylation, looked at different expression metrics, proteomics, chromatin. We looked at telomere length, immune profiling, cytokines, metabolomics, microbiome, cognition, and vasculature. We want to look at all these features. And in this sort of multi-omic analysis of human spaceflight, one of the things that we published a couple of years ago, is that basically telomeres got longer. We saw, this is from one of the summary figures, some features of the metabolome change, cognitive metrics change in terms of his speed. We saw these clusters of biology that kind of went up and down in concert with each other. But one of the features of the study that's not as widely discussed, because in some cases it was kind of, it was unexpected, is that we did see shifts in the epigenome in Mark and Scott when he went in space. So this is data from Scott in space. You can see for these regions, these types of genes, was it T cell differentiation or platelet aggregation? What were some of the genes that were activated that were differentially methylated before and after flight? This is early versus late flight, early and late versus post flight. You can see differences between them. And some of them make sense, like response to radiation or in general immune activation. So those made some sense. But what's interesting is we've done follow-up work since then on a lot more deeper sequencing of these samples and more time points. And here's what's kind of surprising. If you look at in-flight, again, this is Scott in space and Mark here on Earth. If you look at the percent methylation difference, which often 25% is where you put a threshold. And if you look at in-flight versus pre-flight or post versus pre, really by almost any metric, it looks like Scott, who went to space for a year, across his whole genome, he was actually, you can kind of see it here. It's relatively flat if you look at the whole genome for just similarity metric. But if you look at how many differentially methylated cytosines we see, or DMCs, it was actually much harder to be on Earth for a year, at least for the epigenome, than it was to be in space. And so we thought about this a while, and we thought, well, and this is not yet published, but it's very interesting to think like, well, why is that? How could that be? We mostly think it's because, his regimen in flight was actually much more regulated than it is on Earth. So, he'd get up, he'd work out almost every day, really, well organized schedule, would get usually a pretty solid night's sleep once his body got used to being up in zero gravity again. Whereas Mark is on Earth, it's actually harder to get blood samples from the guy on Earth than it was from the guy in space. Kind of makes sense. The guy in space can't go anywhere. But we control his schedule. But the guy on Earth is having nachos. He's having margaritas. He's now thinking about running for Senate at this point, maybe when we're collecting some of these samples. So he experienced a lot more differences being on Earth for a year than what we saw in flight. So what's interesting is, those are a lot more different, but did they change other things like his epigenetic clock, for example? Did that matter? And so we did look at this. For the most part, it's pretty stable. We can see here across CD4, CD8 cells, there is some fluctuation of 1 or 2 years that deviate from what the expected is. And in some cases, their CD8 cells are a little bit older than you would expect by chance. But for the most part, they're pretty noisy and within the range of each other. They're fairly stable. It certainly didn't really dramatically increase or decrease. If anything, it's kind of some good news because at least they didn't get older epigenetically, and neither one of them did over the course of the mission. But we did want to jump back to some other uses of the long reads and jump back to some other kind of questions about epigenetics and gene regulation. In particular, one of the other questions that came up in the early studies is that telomeres getting longer. We want to see if we could use nanopore reads to look at that as well. And so we want to look at this not only for the 1-year mission, but we also looked at people who climbed Mount Everest. And we could see here that actually, 11 other astronauts and people on Mount Everest all get longer telomeres. But when we break out the long reads, now that we know that they're working for methylcytosine, what else can you do with these long reads? What we want to see as well, could we validate telomere length with them? And so some of the early work that was done actually by Dr. Fuchs, as well as Kirill Grigoriev in the lab, was to see, if you try and map them, some of our first mapping attempts really failed. This is the mapping quality, which you can see a mapping quality close to 0 is about as good as you could imagine, not that good, right? So you can see it wasn't really working there. The other weird thing that happened, and this is, a bit of a cautionary tale on the nanopore data, is that while we're extremely elated at its recent efficacy for methylcytosine calling, it looks like it's doing great. We have seen in the highly repetitive areas of the genome, if you try to look at TTA-GGG, which is the canonical telomeric repeat, this is the different versions of the software and called on the same raw data, the proportion of hits you get to telomeres changes. Again, same raw data, just 3 different versions of the base caller, just calling the bases, completely failed in some cases. So we knew unquestionably these were not the telomeres that we were looking for. We felt like really dumb stormtroopers didn't know what to do. So what we did is developed with Kirill and Dr. Fuchs and soon to be Dr. Bezdin and Dr. Grigoriev, as something called EdgeCase, which is actually this just got accepted in the Genome Research, and this will be coming out soon, is a tool set that takes these long reads and says, well, what else can we get out of them? And can we map them to find different motifs, get proper mapping to the telomeres and subtelomeric regions, and see what we can pick up. And so in particular, what we can now see with sort of mapping to the edge of the genome and then the extended reference genome here in gray is we can pick up quite well these TTA-GGG repeat. You can see it here. It's actually quite striking. These are 40, 50, 60 kilobase long reads. You get no telomere, no telomere, no telomere. Suddenly, boom, you can very readily and easily see this density of the repeats of TTA-GGG. But we did see some funky things. There's other non-canonical repeats like TGA-GGG that look like they were being called. So we compared this to PacBio data, to the hi-fi reads, and we could see some of the same things. PacBio data is a little bit cleaner in these repeat regions where you get nothing, 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 and then boom, you are clearly in the TTA-GGG telomeric repeat, which we can really readily see. And then we use this to infer the average length of the reads from Nanopore data, which we could confirm that the telomeres were indeed longer. But to do a little bit of a deep dive on the Nanopore reads, what else you're getting from them, and this is, looking at the diversity of what's present. When we take these long reads and essentially look at the telomeric regions or subtelomeric regions, you can see you can read out into the telomeres and kind of calculate their length. And what we've observed is kind of really intriguing. As we look, for example, here we look at the Genome in a bottle samples, the Chinese and Oscar-Johnny trio. We can see here these trios, like, did they inherit them the same way? What's their relative distance, the Levenstein distance? And if you look over here, this is green is the canonical telomeric repeat, TTA-GGG. Yellow is the non-canonical one, and this is background, and this is the start of the telomere. And you can see up here, this one is pretty particular to the Chinese trio. It's a non-canonical kind of telomeric repeat that we only see here, that we don't see elsewhere, but these also have their own telomeric variants that we can pick up in these populations. The other kind of cool thing is we can pick up, these are the intra-population, there's also intra-population variation, and some clustering. So we get a lot of these, peculiar kinds of variation in the telomeres, which is exciting because it means we can suddenly see new kinds of genetic variation that we were blind to before. And we also, from these regions, we can see they're generally very hypermethylated. So we can see different degrees of methylation or base modification also in these telomeric repeats. So again, this is coming out in Genome Research in a few months, very new publication. So I'll close with, I mentioned some other variants. What about those other variants? As a quick comment on epigenetics as well, is that, for example, in Scott, we saw when he was in flight, his CHIP data, or the mutation of TET2, decreased in flight and seemed to be staying low. But we did see when he got back to Earth that the TET2 variant kept going up. And so this means that in his DNA, take all the DNA of his cells, it looks like it was coming back. But if you look at Mark on the ground, it was mostly stable, but one mutant went up as well. And then when we looked essentially at different cell types, we say, well, is it present in CD4, CD8 cells, or is it in B cells, CD19 cells? We could see here that it was in most cell types, but is at lower levels for some of the sorted cell populations, but it was picked up. And then we use a 10x genomics linked reads and RNA sequencing to look at the difference between DNA and RNA. And this gets to the epigenetic question of, okay, I see mutations in DNA. I want to know, do they show up, essentially in the RNA? And we use the linked read protocol from 10x Genomics to do both deep sequencing as well as linked read sequencing. And then looked at the RNA-seq data as well. We actually saw very little evidence of it in the DNA, but we could pick it up in bulk blood. But then we looked at the RNA sequencing, it looks like for sure the expressivity of the mutated alleles, the RNA here, looks like it was coming from different alleles. If you look here, this is the RNA sequencing here versus here, and it looks like they're showing up, on two different alleles. So it seems to be maybe there are two independent mutations, but it's important to point out that just seeing a mutation in DNA doesn't tell you how much it's actually expressed. What is the expressivity of a mutated allele? You need to have both sets of data to understand the epigenetics or the gene regulation behind that feature. And so one last closing thought on what we see in astronauts versus other cancer patients who get beam radiation therapy is that this is kind of mid to low range of the kind of variant allele frequency changes that you might see. So in this plot here, this is for patients that were untreated in gray or that had radiation therapy in blue, or the astronauts. And you can see here, if you just look over time, people will have a shift in their variant allele frequency for DNMT3A mutants or TET2 mutants. You can see some go down, some go up. Same over here, some go down, some go up. But for the most part, some of the really big changes we could see would occur more when people had cancer patients that had radiation therapy. But you can see you always have to put the context of a VAF change relative to the patient's prior state, like where were they and how does this compare in general to other patients. You can see kind of this result here. Okay, so it means that Scott Kelly got a little bit taller and younger, although the taller went away, and the younger, looks like we're still keeping an eye on him, but as overall good news. I'll close with one last thought about other applications where I think proper epigenetics and multi-omics integration will be useful. Is when we just tweak the epigenome. There are a variety of tools and methods that are now coming online over the past 4 or 5 years, where you can actually basically embed a TET enzyme or a DNA methyltransferase to a Cas9 and add a guide RNA, and then basically target, make your double-stranded break, and have an epigenome editing effector molecule, and use the CRISPR system to also do epigenome editing. And there's a great review paper here by Charlie Gershbach that kind of shows you can help guide cell differentiation. You can look at epigenetic regulation, guide screening. You can create new regulatory networks or study gene regulation. Like Rudolf Janisch has done a great study for Fragile X syndrome, where he reprogrammed Fragile X by DNA methylation to remove the phenotype. So these methods really open up a lot of interesting directions also for not just genome editing, but epigenome editing as well. And it's even led to really interesting projects like with DARPA, where they have a project calling, and also some projects in our lab, where you're trying to do a preemptive expression of protective alleles. You want to actually activate genes when you need them. For example, if you're going into an area with high radiation, could you activate your genes for DNA repair and then turn them back off again later? And so this gives you a really interesting sort of view of what the military is thinking about, as well as people doing therapeutic applications of, let's just, tweak the epigenome and then actually put it back later. But to do so, we will need really good measurements of it, as we just described here, and some of these benchmarking and standards will help in that regard, we think. And one last place where you can use epigenome editing, someday maybe you could do it so you could activate people when they land on Mars or other planets. And we just I'm just a shameless plug. I have a book coming out next week, so I thought I'd tell everyone about it because where I talk a lot about epigenome and genome editing and measurement. Okay, I want to thank everyone in the lab who made this possible, and this includes Scott Kelly, as well as other members of the expedition. This is a picture, an older picture of the lab. Everyone who made this work possible. A more recent picture where we all got together post-vaccination and are now starting to actually see each other again, which is a lot of fun. And really big props to Dr. Fuchs here, who's visualized here, looking like someone just grabbed his leg. I'm not sure there, but this is all of us hanging out just recently. So thanks again also to the NIH, the FDA for some of the funding of this work, as well as World Quantum Gates Foundation, and the NIH, of course, as always. And finally, thanks to everyone in the lab who makes this possible, as well as a phalanx of friends, collaborators, and colleagues, especially here in the MAQC and SAQC. Thank you so much. And I will take questions for the hour.
Okay, well, Chris, that was a tour de force as usual across the spectrum of epigenetic and related analysis. I have some questions of my own. So one was, I know you focused on a lot of the sequence-based methods looking at reproducibility, but I also know you mentioned some of the array-based methods for measuring methylation. And I wanted to find out, in your general impression, how did the two compare, and what are the, for some that maybe are just getting at epigenetics, what are the, the big advantages, disadvantages in looking at sequencing versus, arrays for looking at different aspects of epigenetics?
Great question. So actually, I am remiss. I have slides on that, but didn't include them. So let me pull it. Can you see my screen now?
Yes.
So we did. What's interesting is and this will be in the updated paper. This is basically the draft of the whole figure. This will be an entire figure. Is just actually. There's multiple facets of the study that I should have included here. One is which aligner works best, frankly, when you have the methylation data, when you're taking raw sequence data and do the alignment. And then also if you have array data, what do you do for normalization methods, and how does this change your beta values or your methylation value when you use the microarrays, the EPIC arrays? And so we could see here is, you want to basically try and get, good concordance between the epigenome across normalization pipelines. And so we could see here, it's kind of like what's been published for other MAQC or SEQC studies, that sometimes the normalization methods that you have, don't always make things better. They might change things only slightly, whereas other ones dramatically improve, what's the concordance. And so you're seeing much more concordance. So for example, these different post-processing and normalization pipelines can, you're looking for more red as the pipe combinations we most strongly recommend for microarray data. And then also we looked at what's the concordance between the higher and low varying sites. And, you can see here again that there's some of these different methods, and these are, there might be other methods out there. These are the ones that we found are most commonly used in the field. for the microarray normalization. And also we can see, some do better than others, but overall the concordance is pretty high between labs. And this has been published by other groups, so it's not shocking, but it's always just nice to see that, there is some, overall high correlation, but there's also some bad actors. There are some specific SNPs you can see here, different sites on the array that are noisier than others, right? So this has been true for arrays since the dawn of arrays, is that whether it's genotyping or methylation, there's always going to be some probes that are a bit noisier than others, and that these are basically these high varying CPG sites, even if you account for SNPs. And so we've put these are kind of like we're calling them almost like the blacklist, or the tread carefully list, or the kind of in the genome sequencing talk you'll hear next week, we call them classes of evil, or just really bad performing sites. And so this is a bit on, let me go back to this is what's driving some of the variation. I'll jump. So this is the array data. For the differences between also the platforms, like what's driving the variance, you can see here that most of it's driven by cell line, and then a little bit by assay, and then the residual variation, depends on which platform you're using, but Nanopore does have a bit more of an assay specific variance factor you can see here. And then, so this is a little bit on the source of variation. One last thing while I'm sharing my screen, I believe you can see my screen here still, right? And we're in the middle of finalizing this, so I will show you. Actually, I think I just downloaded it. Hold on, I'll show you. I was looking at this last night. Okay, yep, here it is indeed. So, and can you still see my screen?
Yes, we can still see it.
Great. So this is one of those key questions of, this is figure 2. Yeah, so figure 1 is, let me just highlight this, is there's a variety of tools for doing the alignment. So a lot of them are BWMF, or doing BizSeq. I don't know if you've heard of, actually you can see what's being updated, you can see here, but Bismarck, Bitmapper, ARBS, BSSeeker, or BWMF. These are common tools for doing the alignment. So one of the other questions we have that's in the updated paper is, how many reads are primary mapping, or multi-mapped, or duplicated reads, or unmapped reads? And BWMF generally does a really good job. It maps more, but also gets more multi-map. But generally, when it does get mapping, you can see what's the percentage it's mapping. It's generally mapping more across each one of these platforms. You're getting more coverage of your CPGs, you can see with BWMF. And if you look at what are you seeing when you do the alignments, are you getting different parts of the genome? They're mostly pretty consistent between each one of these aligners. And so, another part of this paper that's not coming out is, we've looked at a variety of ways to do the alignment, the very first step of the analysis, and found, basically can say here so far, at least with these versions and these data, that BWMF looks like it's probably the best way to go forward.
Okay. Okay, well, great. So
I realized I should have included those. Those are like really great analyses that we just finished, that John just finished with the group, but like it was, we have them in there. I should have included them.
Oh, that's great. So my next question was related to the reference sample that you mentioned that you used, which were from Genom in a bottle. So can we assume that with the publication of the paper and related information that just like Genom in a bottle has published variants that they have seen in high confidence regions or whatever, would there be sort of an analogy related to where you'll publish methylation related characteristics maybe in high confidence regions for those same samples? So is that part of the plan to release that?
Yep. For each one of the CPGs, there's these variance metrics of how noisy are the sites, and we know it really well for the DNA sequencing, because that's what we've been building out for the NGS study, is what across all platforms. And again, there's a quick plug for a really great talk next week on basically all the existing platforms that are on the market today, including Genapsys, which just released. And we actually just got last week some cool MPS data from MGI, BGI, which I don't know if it'll make it into the paper because it's already been resubmitted to Nature Biotech, but I don't know if it'll be done by next week. But anyway, we'll have some of that data at some point we'll talk about. But in there is a list of the classes of evil, the really hard to map areas of the genome where we see a lot of variation. We'll include the same thing here, which is basically a large supplemental table of the areas which you can assign a prior probability of more noise. So, just to kind of, be more careful in those areas.
Okay. And then I was intrigued by the telomeric analysis that you were doing, where you showed these these alternative repeating patterns. Instead of the main one you were looking for, you found the presence of a competing repeating pattern that was maybe highly even specific either to a population group or to particular chromosomes or whatever, but have you had a chance to understand the functional significance? In other words, is it just like an acceptable alternative allele, or is there some functional significance potentially to those alternative repeat patterns? Yeah, the TGA GGG one versus the TT.
Yeah, so great question. And this is something, we just we don't know yet. The short answer is it's not clear. But what's really just intriguing is, the telomere lengths have been linked to aging, also been linked to, essentially cancer cells often can keep their chromosomes longer. What we're in the process of doing now is getting more and more samples that have these ultra long reads so that we can actually read into the telomere. We just don't know. We started with these samples because, of course, we know we can get the mapping right. We know what we're looking for. But this has really opened up a new area of how much do we see weird non-canonical sites showing up in cancer samples, or as a disease progresses, can this also be another way to mark which cells you're picking up? And so we just don't know at this point, but it gets us a window into a place of the genome we just really haven't been able to see before.
Okay, we've got a question here. So one about epigenetics and cancer. So I think you illustrated a couple of examples like IDH1, IDH2, where epigenetics plays maybe a critical role related to certain cancers. Are there other main either epigenetic groupings or genes where you know of where epigenetics plays a role maybe as a driver in cancer, or either as, if not a driver, maybe as an inhibitor for say DNA repair or something like that?
The best one of the ones I just described here is the IDH mutant samples, but we'll see also in other and not just in leukemia, but the most notable example outside of leukemia is in glioblastoma has a hypermethylator phenotype where you see it looks like someone took a essentially a hose of methylation and sprayed the genome. It's a whole genome gets methylated, which of course often turns down gene expression and generally dysregulates a lot of the expression for most genes. So it is that can be from DNMT mutations, so DNA methyltransferase. That's one other case where we can see it's a real driver. What's interesting is the question is it genetic or epigenetic? Like the mutation is genetic, but then it's in an epigenetic modifier, which is what actually ends up leading to the challenge or the problems. But that's another classic gene that's been linked. And then also there are HDAC mutations as well, histone deacetylases that are common in some cancers. And that's more at the structure of the histones and control of how the DNA is packaged, where you see that can drive the cancer. There's even been some papers, we've published a few papers where just variants of histones like H2A, but H2A 0.2 or 0.3, slight weird variations of histones can sometimes show up in cancers where just the protein subunit has changed and is an indicator, at least a biomarker of the cancer.
Okay. All right. Well, that's all the questions I think we have right now. At least I haven't seen any additional questions, but Chris, we've certainly enjoyed your presentation. As always, learn a lot when I see your presentation. I'm always inspired to do more in epigenetics because you do a great job of presenting, how important it is as another omics aspect to be aware of whenever you're looking at a full complement of the different aspects that impact cells and disease. So thank you very much.